Bismillahirrahmanirrahim. Lecture number 3 is the modification of polynucleotides. Means different modified nucleotides they are involved in the DNA, present in the DNA and also in the RNA, and they how this modification they are helpful for the different functioning of the DNA and also in different types of RNA. For example, the modified nucleotide, that is the 5-methyl cytosine. This 5-methyl cytosine, it is present both in bacteria as well as the human DNA. Methylation basically it turns the DNA or the gene into inactive form. If there is a greater methylation, it means that gene it will be inactive, or this portion of the DNA it will be inactively behave as a gene. For the activation of this gene, this methylation should be restricted, and it will be converted into the acetylation, and not on the same place but at different places, and so as to convert this inactive gene into the active gene. This is also known as the chromatin remodeling. Methylation also in bacteria, it is helpful to prevent the bacterial DNA from the foreign DNA, as when foreign DNA it will be entered into the bacterium just like a virus, then there on the bacterial restriction endonuclease it will be break these foreign DNA, but it will not break its own DNA. So this bacterial DNA it will be prevented due to this methylation, because these restriction endonucleases they recognize, they identify their own DNA as the methyl DNA. So this methylation is also helpful, and this methylation is present, the methylated nucleotides they are present in both in bacterium as well as in human DNA. And other methylation or modified nucleotide in the polymer, that is the 5-hydroxymethyl cytosine. These methylation occur in bacterium as well as in the viral nucleic acids. 5-hydroxymethylation occur in bacterium as well as in the viral nucleic acid. Similarly, the mono and the di N-methylated adenine and guanine, this is also present in the mammalian messenger RNA for the stability of the messenger RNA, and also for the proper functioning of the messenger RNA. Methylation is also present in at RNA, just like a messenger RNA. These are the different modified nitrogenous bases, and they incorporated into the DNA as well as in the different types of RNA, and helpful in the proper functioning of the DNA or the RNA. For example, the 5-methyl cytosine. Here we number this pyrimidine base as 1, 2, 3, 4, and 5. This is the clockwise numbering in pyrimidine. And here the 5 position, it contains the methyl. This one is the whole structure is the cytosine, and at the 5th position there is a methyl residue. So it is known as the 5-methyl cytosine. This 5-methyl cytosine it is present in the mammalian DNA as well as in the bacteria. 5-hydroxymethyl cytosine, here the one of the hydrogen it is replaced by the hydroxyl group, and this one make this 5-hydroxyl hydroxymethyl cytosine. This 5-hydroxymethyl cytosine, this is present in the viral DNA as well as in the bacterium DNA. Dimethyl amino adenine. Here the exo amino group it contains the 2-methyl residues, so it is known as the dimethyl amino, and the whole structure is the nitrogen base is the adenine. This adenine that contains the dimethyl amino residues, exonucleic dimethyl amino residue, it is present in the messenger RNA. Similarly, the 7-methyl guanine. 7-methyl guanine at the 7th position, the guanosine contain at the 7th position the methyl residue. That is important in the 5 prime, in making the 5 prime cap of the messenger RNA. So these different modified nitrogenous bases also present in the messenger RNA, in the DNA, along with the usual standard nitrogenous bases, that is the adenine, guanine, cytosine, uracil, and thymine. Uracil is present only in the RNA, but the thymine is present only in the DNA. Similarly, the body also contains different modified nitrogenous bases due to the metabolite. These are the metabolic products of the purine: hypoxanthine, xanthine, and uric acid. They are the derivatives of purine, also known as the derivatives. They are achieved, or they are formed after the degradation of purine. Purine metabolism, they form the hypoxanthine, xanthine, and uric acid. Hypoxanthine, that is, it is the 6-oxopurine. The nitrogenous base, it is purine, and the 6 also, if we number this, 1, 2, 3, 4, 5, 6, 7, 8, and 9. This is the anti-clockwise numbering, and the 6 also purine, it forms the hypoxanthine. Similarly, 2,6-dioxopurine, 2,6, that is, even number 1, 2, 2, 3, 4, 5, 6, 2,6-dioxopurine. It is also a modified purine base present in the body. And the uric acid basically, when it is formed, it means 2,6,8 trioxopurine. This one is the purine derivative, trioxopurine, and it is hyperuricemia. It causes the hyperuricemia, that is excess of uric acid. It will be accumulate in our soft tissues of the joints, and they form the uric crystals, and it will give rise to the arthritis. So one of the cause of the arthritis is the accumulation of this uric acid. This uric acid, it is the derivative of purine, 2,6,8 trioxo. This one is the trioxopurine. Another derivative of xanthine, that is the trimethyl xanthine, which is also known as the caffeine present in the coffee. It is the oriented modified nitrogenous base, which is the derivative of the xanthine. Here it is the trimethyl xanthine. Trimethyl xanthine 1 and 3 and 7 position. The methylation take place at 1, 3, and 7 position. The trimethyl xanthine, this trimethyl xanthine, it is present in the coffee. And another, this is the trimethyl, but the dimethyl xanthine, just like a theophylline and theobromine, it is also present. The structure of theobromine, that is, the methylation takes place at the 3 and 7 position instead of at 1 position. And similarly, the theophylline, the methylation takes place at 1 and 3 position instead of at 7 position. The theophylline, it is present in the tea, and this theophylline, it is the bronchodilator. The synthetic nucleotides, modified nucleotides, they are naturally occurring, present in our body, as present in other DNA. In humans, also the modified nitrogen species, they are present in the bacterium and also in the virus. But there are the synthetic nucleotides. They are known as the analogues, and they are used in the chemotherapy. Very important subject for the modified or the synthetic nucleotides analogues in chemotherapy or in the medicine. These thin synthetic analogues of purine, pyrimidines, nucleosides, and heterocyclic ring. Heterocyclic, maybe modification occur at the heterocyclic ring, or maybe at the sugar moiety, or maybe on both sides, so as to form the synthetic nucleotide analogues. Analogues mean they resemble to their original structures, but there are some little bit differences. So they are the derivatives basically, and numerous applications in the clinical chemistry. The toxic effects is that they are basically they are synthesized on selective basis. They inhibit the DNA or RNA on the microbes, or may inhibit the DNA of the parent, mean the host or the human, so as to stop the cell growth. The toxic effect is maybe the effect on the enzymes, which these enzymes they are used in the nucleic acid synthesis, or may they incorporate into the nucleic acid. And nucleic acid mean they are may form a structure, they are analog structure of the nitrogenous bases, and when these nitrogenous bases they incorporate into the DNA, so they disturb the base pairing. So on both these actions, one is to may inhibit the enzymes which are used in the nucleic acid synthesis, whether in DNA or RNA, or may incorporate the analog into the nucleic acid, whether in the DNA or RNA, or which results in the disruption of the base pairing. Oncologists employ 5-fluoro or 5-iodouracil or 3-deoxy. There are different types of analogs: 5-fluorouracil, 5-iodouracil, or 3-deoxyuridine, 6-thioguanine, 6-mercaptopurine, 5 or 6-azoridine. These are different. These are the name of different synthetic analogs. Or 5 or 6 azacitidine, or 8 azaguanine. They incorporate into the DNA prior to the cell division, and during the cell division, when replication take place, they stop the replication. At this stage, they will, if these analogs they are used in the DNA, then they will be stop further replication. They will stop the replication of DNA, because the analogs are present, and there is no complementary or complementary basis in the cell, so it will stop at this position. Purine analog also present, just like allopurinol, which is used in the hyperuricemia, gout, or inhibit purine biosynthesis and xanthine oxidase activity. Allopurinol, the function of the allopurinol, it inhibit the enzyme xanthine oxidase. This xanthine oxidase, it is used to synthesize the xanthine as well as the uric acid. So when it inhibit this enzyme, it will block the synthesis of mm, uh, xanthine and uric acid, and so it will treat the hyperuricemia. These are the structures of the synthetic analogues, whether they are pyrimidine or purine. For example, 5-iodo 2'-deoxyuridine. 2' mean deoxyuridine at the ribose sugars. So at 2' position on the ribose sugar, there is the deoxy position. And 5-iodo at the fifth position, 1, 2, 3, 4, 5. Fifth position, there is a iodo group present. 5-iodo 2'-deoxyuridine. 5-fluorouracil. Instead of iodo, there is the fluoro group. 5-fluorouracil. These nucleotides analogues they incorporate when into the DNA. They will stop the further replication of the cell. Azoridine. Azoridine means there is a nitrogen to nitrogen. This one is the uridine, and nitrogen to nitrogen at the sixth position, nitrogen to nitrogen bond, and addition of the nitrogen at this position. And azaguanine at the eighth position, and again there is a nitrogen. There is a addition of nitrogen. Similarly, mercaptopurine SH group incorporated at the sixth position. This SH group known as the mercapto, and this is the purine derivatives of mercaptopurine. Thioguanine. The SH group is present at the sixth position. This is the sixth thioguanine. And allopurinol, as I have already told you, that it is used for to inhibit the xanthine oxidase for to treat the hyperuricemia. So allopurinol here is this one is the derivative of adenine or purine degradation. It is found from the purine degradation. The xanthine as well as the uric acid, they are formed from the purine degradation, and the xanthine oxidase, this structure is just like a xanthine, and the xanthine oxidase, it is inhibited its structure is just like a hypoxanthine, and xanthine oxidase use this substrate, and there is no formation of this one is the competitive inhibitor, so no formation of the product. This one is the hypoxanthine. Instead of using this hypoxanthine allopurinol, it will inhibit this xanthine oxidase, and there is no formation of xanthine. Xanthine oxidase, it shows its function at both places. Here hypoxanthine being converted to xanthine, and again this enzyme will act on the xanthine as a substrate and form the uric acid, so it will stop this at this position. Cytarabine, which is used in the chemotherapy of the cancer, and azathioprine, this is catabolized to the 6-mercaptopurine during the organ transplantation to suppress the immunological rejection of the organs. So these are the different analogues which are used for different purposes. This is the structure of cytarabine. Here the same cytosine residue in the nitrogen space, but the difference is here the ribose sugar that contains the hydroxyl group. Here the arabinose, instead of the ribose sugar, the arabinose is used. Similarly, azathioprine, the structure of azathioprine, that is the nitrogen base, it is linked through this thio bond. So different. This is the used for the during the organ transplantation. 5-fluorouracil, it acts in several ways, but it is principally as the thymidylate synthase inhibitor. This is the enzyme. The use of the 5-fluorouracil is it inhibits the enzyme thymidylate synthase, which is used for the synthesis of thymine during the DNA replication. So it stops its replication during the chemotherapy. And pyrimidine nucleosides, they are used mainly in the treatment of the leukemia, especially acute non-lymphoblastic leukemia. Cytarabine, it is an anti-metabolite, anti-neoplastic agent, and it inhibits the synthesis of DNA. And also its actions are specified for the S phase of the cell cycle. If the question is that what is the function of the synthetic analogues, then you can write all of these functions. It also has the antiviral as well as the immunosuppressant properties. This is the role of the cytarabine in chemotherapy. Similarly, there are the research tools just like triphosphate analogues, which also used as a research tool, and they are the characteristics of these research tools are that they are synthetic non-hydrolyzable analogues of nucleoside triphosphate, and they also affect the transfer of the phosphoryl group, and also the also affects the allosteric nucleoside binding sites on the regulated enzymes. These are the synthetic derivatives of nucleoside triphosphate. Um, This one is the parent nucleoside triphosphate. The three residues of phosphate, they are linked to the ribose sugars. But in case of beta and gamma methylene derivatives, here the methylene bridge is present between the two phosphate group and or the imino derivative that is present. So these are used for the tools, and they act as a synthetic tools.